Hello and welcome to this webinar organized by Alphalyse. So we have this series of webinars on CMC related topics, and today with me I have Margarita from Genmab. So Margarita will share her experience from a project at Genmab. So before we start Margarita's presentation, I would like to welcome you again.
Thank you.
So, and to the audience, we have about a little more than 200 sign-ups for this webinar. So there's a huge interest from other CMC managers and analytical scientists and project leaders. So we are very happy to facilitate this audience. So if you have any questions during Margarita's talk, then please type them in the chat, and then we will come back to them after the presentation. The presentation will be about half an hour approximately, followed by 10-15 minutes discussion. So with that, I would like to introduce again Margarita. So Margarita is an expert in HCP analysis. She is, I'll read your bio here, Margarita, senior subject matter and analytical lead in the late manufacturing development at Genmab. Her group is responsible for developing and validating analytical methods, including potency assays and host cell protein assays for monoclonal antibodies. Margarita has a long experience from HCP analysis, also in other companies, including Novo Nordisk. So with that, Margarita, welcome.
Thank you.
So I'm really looking forward to your interesting presentation. So I'll just start the presentation here. Turn screen share on. Window. So with that, please go ahead, Margarita.
Thank you, Ejvind, and thank you very much for the presentation, introduction, and for the opportunity to share Genmab experiences with the control strategy. But before I start, I would like to make a disclaimer. I will present the work performed by Genmab, but along the way, I will give you some of my thoughts and views that are mine. So just a short introduction. I don't know how many of you are familiar with a control strategy. The control strategy it's defined as a list of process inputs and outputs. The outputs are normally what we call CQAs or critical quality attributes, and these outputs need to be under control in order to ensure that the process delivers consistently a product of desired quality. So traditionally, HCPs have been identified as critical quality attributes that need to be controlled, and that's why we normally have a control strategy around HCPs. But this control strategy has different levels of controlling HCPs. Normally, you will have a release testing by a suitable method that demonstrates the process consistency, and you also will have clearance studies to show that most of the HCPs had been removed. So in this, at Genmab, we have established a holistic HCP control strategy that encompasses a set of approach for the selection of CHO HCP reagents, since we are working with CHO host cell proteins and host cell for production of our monoclonal antibodies. Besides the selection of reagents, we perform identification of the HCPs to determine if there are any problematic HCPs present in our drug substance. And besides that, we perform process development and what's the impact on the HCP profile by different process changes, and again, the process control and process consistency demonstration. So probably you already heard my colleague first part of the presentation. Søren make a good presentation in how to select the most appropriate release testing method, including the selection of CHO and anti-HCP ELISA regions, identification of the HCPs, and coverage analysis using ELISA-MS. In this part, I will focus on how to use LC-MS for assessing process changes and demonstrate HCP clearance and document process consistency. I will round out my presentation giving you insights and sharing the experiences and feedback from the authorities in licensing applications in Europe, Japan, and US markets. And maybe some other markets will also be included at the end. I hope that this introduction can give you a slight or at least a good feeling about the things I will be showing. So the agenda once more is I will give you examples of using LC-MS and the experiences and feedback from licensing applications. So going to process development and HCP characterization. So one of the first steps in the holistic controller strategy was to obtain knowledge and understanding of the levels of HCP, but also what type of HCPs were in our mAb production process. And what were the HCPs that were remaining in the bulk drug substance in the process. So for that we use characterization of the HCP profile by LC-MS, and the analysis was performed on in-process samples for clarified harvest, a chromatographic step 1, a chromatographic step 2, and the bulk mAb product. So here you can see that we performed the LC-MS analysis, and here are the identified HCPs plotted in a 2D plot where we have the PI on one scale and the molecular weight in the other scale. As you can see, there are many HCPs in what we call the 4 quadrants of a 2D plot. What we can also see is that we obtained approximately 800 HCPs were shown in the clarified harvest. About 72 of them, the ones marked in blue here, were identified in the first chromatograph after the first chromatographic step. And then about 4 were identified in the second chromatographic step, that the the green ones. And about 3 were identified at last in the bulk sample. You cannot see them all here, but they are around PI of around 8, and it's consistent with our products PI. It's around also the same area. So there were only 3 proteins left in the bulk. One of them was identified actually as a residual protein A from the chromatographic column 1, and a second of the third was a problematic HCP. So we tried to investigate a little bit further on that, but we also could see that we have a nice reduction through the process. One thing I would like to say is that knowing the PI or molecular weights of the proteins, HCP proteins, it aids in developing a purification process in order to remove them. So we did some process changes, and of course, every time you do a process change, you have to compare the output of the process, not just the quality attributes, the normal ones like high molecular weights and low molecular weights and so on, but we also apply the concept of comparability to the HCPs. So the process changes we introduced from what we call process 1 to process 2 were a change in the master cell bank to use a working cell bank, change in manufacturing site. We also, because of the change in manufacturing site, we did multiple process adaptations and also did scale up. So will also result in some chromatographic optimizations. Again, we analyze different process steps with LC-MS to identify what type of HCPs were in both processes. And we analyze, again, clarify harvest, the chromatographic 1 output, the chromatography 2 output, and the bulk drug substance. Sorry. So in this comparability analysis, we showed that in process 1 we actually have around 800, as I show you in the slides, the previous slide. And process 2, we actually only had, well, not only, but we had around 400 proteins after harvest. We also could compare the chromatographic states against each other, where we had about 80 proteins after first chromatographic step and process 1, and we only have 6 in process 2. And the same chromatographic step 2, we had 6 in process 1 and 2 in process 2. And at the end of the DS sample, in this analysis, we identified 5 HCPs in process 1, and there was only 1 left in process 2. And when we compared the type of HCPs that were in harvest, we could see that most of the HCPs, around 75%, were the same HCPs in both processes. And however, there were about 100 new HCPs that were not present in the process 1. When we look at the first chromatographic step, we had about 4 proteins that were the same, and 2 that were new ones that we didn't see in the first process. So in this comparability assessment, we could determine that we had a good reduction in process 2. It wasn't a worse process than process 1, and that we only had 1 protein left in the TS bulk. So that was the process 2 was the one that we will be going commercial. But before going commercial, we actually had to have a process change more. And that what we did was assessment of the harvest step. So due to COVID-19 and liver issues, we have some issues having a specific filter delivered. So we needed to assess what was the impact of this harvest stage filtration regarding the HCP impurities. Will they have an impact or not? So the normal process was with 3-stage filter, having 2 depth filters and 1 in-phase filter at the end. This in-phase filter was shown to be if I go back to previous slides, So the reason why we had a reduction in the number of HCP proteins in harvest was due to this implementation of this in-phase filter. That was very good to remove HCP. So that was our commercial process, or intended commercial process, using a three-stage filtration. But due to the COVID-19, we couldn't get an extra in-phase filter, so we only went with two the filtration. So we did again the characterization of the HCPs, and we look again at the pre-harvest sample from the bioreactor before the depth filtration, and then the clarified harvest, and again chromatography 1 and 2. So here are the results. If we look here in the sample prior harvest, we of course have more proteins that at the harvest stage. So we had around 900 proteins, and using the standard three-stage filter, we had around less than 600 proteins left, while when we used only the two-stage filter, we have about 700 proteins left. What we could also see is that independently of how many proteins were at the start, the chromatographic step 1 could reduce almost all the proteins to around 20. And at the chromatographic step 2, we could also reduce all the proteins to only 1. So what we conclude it was that the two-stage filter work. It was a little bit worse. I think it was could only reduce 24% as compared to 37% of the three-stage filter. But because of the process, the subsequent process tests were very robust and were very good at removing the remaining HCPs. So it didn't matter that we have more at the beginning. We at the end end up with the same one protein. So in conclusion, with this information, we could actually show that the both harvest filter setups were equivalent and could be used, and we showed this in our PLA. So another way we can use or gain knowledge about the HCPs was to show or demonstrate consistent HCP clearance at commercial scale. So that's one of the most traditional holistic control strategy to show clearance through the process, and this is normally shown in the impurity section of filing. So in this case, we use again LC-MS, but we also compare to ELISA results on 4 PPQ runs. Again, we analyze the 4 PPQ runs using the in-process samples. Again, clarify harvest, chromatography 1, and chromatography 2, and at the end, the drug substance or bulk product. With the caveat that actually the PPQ4 was the one that used the alternative filter setup. And here are the results. Sorry for having a very busy slide. I will try to guide you through the slide. So we have the 4 PPQ batches, and on the first column we have the ELISA results. On the second column we have the number of HCPs, and on the third column within each PPQ batch, we have the MS results as a quantitative result. So we can see that we have a lot of HCPs in our last production bioreactor sample. Unfortunately, we didn't actually analyze the same sample by LC-MS. So we only have a few selected samples analyzed by LC-MS. But one thing we can see is that we have, looking at the number of HCPs and also the PPNs by LC-MS, is that we have a robust clearance through all the 4 PPQ batches, resulting in only 1 protein left in the drug substance. Another thing we can compare more is that at the beginning we have around, I would say around 2,700 ppms, and we have a more or less consistent levels also, even it's a little bit lower, but we have also consistent or relatively consistent levels of HCPs measured by LC-MS. We can also see that in PPQ4 we had a higher number, both in ELISA ppms and LC-MS ppms, and number of HCPs as expected. Another thing when comparing ELISA and LC-MS is that at some point with ELISA we cannot detect any more HCPs. They are normally used. They are normally below the LOQ because of the dilution linearity problem. So at some point, the matrix affects the ELISA method. So you have to dilute too much in order to overcome the matrix effects, and then your result lies below LOQ of the method. So that's why they are reported in this way. So yeah, at some point you cannot actually demonstrate clearance because we cannot see any reduction in the next chromatographic steps. Sometimes you can measure something, and sometimes you cannot measure anything. So it's very random results we get with ELISA, whereas with LC-MS we actually see a good reduction. We can see we go from 500 to around 10 to around 1, and it's the same at the end of the filtration and at the bulk substance, drug substance. So we actually have some numbers we can rely on and show demonstrate this clearance. With ELISA we can only say, okay, after some point we cannot measure anymore whatever is in there. So you have difficulty in showing that actually chromatography 1 can reduce, and chromatography 2 can also reduce. You cannot see the effect by just looking at the ELISA numbers. And another thing we can also take from here is that the LC-MS, even though you have a quantitative result, it's not necessarily. I mean, you have a quantitative result, but it's limited by the method. But you also have the ELISA result. It's also limited by the method. So the ELISA result is depending on the affinities of the antibodies and the reference standard, the calibration standard you use. So both numbers, in a way, they are relative to the method it's used to obtain a result. This is also a way to illustrate the same clearance, but we can show that there are mostly the same proteins in all PPQ batches. So we have very good agreement, and we can show very good consistency, because it's the same proteins that you are seeing at consistently in the 4 different batches. So we can see that's the harvest, and then we can see that some of proteins are more in one batch than the other, but consistently they are the same, except for, of course, the some single proteins that are distributed across the 4 batches. But in general, we have a very good consistency of proteins expressed. We also, by the analysis, now that we know what proteins are present, we can also show that the phospholipase B and carboxypeptidase, they were originally present in the harvest samples, but they were consistently removed by the process in all 4 PPQ batches. That knowledge will you never gain with the ELISA method, because then you cannot distinguish between what is a problematic HCP and what it's not. So at the end, I will just give you some feedback from the authorities. So our core submission document contained information about using the commercial kit, as my colleague just showed you in the previous presentation. We went out with we filed our specification method with a commercial kit, and we showed that the coverage analysis by ELISA-MS supported by LC-MS characterization. So having shown this in our core file, the feedback from FDA, they didn't have actually any questions at all on the use of a commercial kit, nor the coverage analysis. They find out there was we had sufficiently justify why the commercial kit was good enough, and the coverage analysis performed by ELISA-MS was also good enough. The same did the UK authorities, MHRA and Japan. They didn't have any questions at all. EMA, initially they didn't question the use of the commercial kit, nor the coverage, and the coverage by ELISA-MS was accepted, but they wanted to have more supporting information, so they asked us to show beside the ELISA-MS the 2D Dutch, and they also asked us to implement a process-specific HCP reference standard instead of the kit standard, unless we could justify the use of the kit reference standard. So we kind of comply with the request and did a 2D Dutch analysis of immune affinity capture HCPs. And then we succeeded in justifying and providing data to show that the key reference standard was basically the same as our HCP process samples, and it wasn't necessary to prepare a process-specific HCP reference material. And then last, Canada asked for development of process-specific HCP directly. So we at that time we weren't good enough to justify the use of a commercial kit, and our partner decided to go ahead and start developing a process-specific HCP ELISA. So conclusions and take-home message. LC-MS is a powerful tool to characterize the HCPs and aid in process development, to identify problematic HCPs, evaluate the process changes impact on the HCP and comparability, and then also to show HCP clearance and process consistency. We have gone further at Genmab, and we actually have initiatives on showing even more or using the LC-MS for more examples that I haven't shown here. But these are, for instance, early identification of the process or the presence of proteases and the development of the purification process in order to remove these proteases. We have also used LC-MS to assess DOE HCP data from a traffic step in process characterization in order to select the appropriate process ranges for that step in order to show the maximum clearance of HCPs. We had also been using LC-MS for bridging of HCP reagents and for risk evaluation of the HCPs present in bulk samples, and also comparison of all drug substance for a specific project in order to show the clinical relevance of the proteins present in the bulk sample, to show that even if there had been a problematic HCP, we can still show that it had not clinical relevance because we have data from our pharmacokinetics and immunogenicity studies, and also we have data for adverse reactions. So in that way, it also helps us in setting specification limits for the HCPs. I think with that, I conclude my presentation. I hope you could get some input, and my experiences or our Genmab experiences will inspire you to do more LC-MS. Thank you.
Thank you very much, Margarita, for a very nice presentation. Thank you for sharing your experience on this. So I'll just skip your presentation and go into camera mode. So very nice presentation. I think the data you show here is very comprehensive strategy, where we have used both ELISA and mass spectrometry in combination throughout the project, and even managed to get the product on market. So I think that's the case most companies hope to get the product on the market. So we have a number of questions where we can discuss some of the details of your presentation. So let me just read here. So many companies are also developing mAbs, and maybe they're using commercial kits early on. So at what stage would you suggest to use mass spectrometry?
I would suggest to use mass spectrometry from the start because it will aid you in showing that the commercial kit is good enough or bad enough, depends. But it will help you to understand what's happening with your process.
So when you say from the start, is that already before you go into the clinic for phase 1, or?
I would say, well, maybe not before at phase 1, but surely after, at least when you start doing process changes. But it's important to know if you have any problematic proteins, because I haven't shown it, but from my company, I know what we have a single project where we actually have a protease that it's degrading the product. So you need to do something. We can manage with a lower yield of product for phase 1 because we had to implement purification strategies, and that resulted in a very low yield of product. So you have to go with phase 1 with what you have. But knowing what we have now, we know what protease it is. We can actually do process changes to try to improve the yield and try to reduce the degradation of our product.
So one concern we hear from some people is not to use mass spectrometry. They're concerned what they might find. So what do you do with the information when you have the list of proteins? So what if you find the potential problematic in there? How do you deal with the information? What do you do about it?
Act on it. I mean, I think the first thing you always do with whatever information you get is to do a risk assessment. You calculate, you evaluate the risk, you assess the risk, and find, okay, is it something we can live with? If it's a protease that is degrading the product on the way, but it's not present at the end of in the drug substance, so then maybe okay, it's a yield, it's a business risk because you get out with less than what you started with. But if it's a protein that will continue to degrade your product during stability, then it's something else. Then you have a shelf life of your product that is much shorter than what you normally will give 3 or 5 years to an antibody product. So maybe you have only 12 months stability data. So you need to evaluate all the risk and pros and cons in order to make them the most informed decision on what to do next.
So these problems, are they theoretical? Is just because we have mass spectrometry now we can see the proteins? Or how often do you experience that there are problematic HCPs in a product?
Well, I must admit, in old times when we didn't have LC-MS, we were kind of blind. It was the ELISA result. It was a black box. You didn't know what was happening. Sometimes you had other controls in your process that or other indicatives in your process that you had a problem, but you couldn't point out what was the problem. Now, with the information we have, now we can point out what is the problem and act on it. Otherwise, I think from my previous experience, we have a problem with HCP that it took a really long time to find out what was the issue, and then maybe you take several years in order to find the cause, where with LC-MS you can find it in less than I don't know how fast can you be, 1 month, 2 months. So it's a lot of resources you spend not knowing what is happening.
So you're trying to guess what is the root cause of the problem.
And you only have indications from maybe other analytical methods.
So when we look into the future and the crystal ball, what's going to happen with HCP analysis? And there's this new USP chapter 1132.1 that's going to be final this summer, we hope. So how do you see the future for HCP analytics for mAb projects?
I think the people are not yet ready. I mean, the authorities are definitely not yet ready. But I can see a progressive approach where at the beginning you start showing data from both ELISA and LC-MS, like a kind of hybrid approach. But my vision and my hopes and thoughts for the future is that we will be able to actually present data without ELISA. I hope in the future we could, instead of filing a specification quantity number for HCPs, we normally have a start with 100 nanograms per milligram, And then on the way, we are reducing to around 10 nanograms per milligram, because the authorities ask us to match whatever it has been in the clinic. So we are often pressed to go down to a quantity number. It's without really knowing what is behind this number. So my vision is that in the future, instead of submitting a release method showing a quantity number and a specification limit, that we could submit a number of HCP proteins we have identified through all our process development, and through PPQ batches we have seen process consistency, and we can see the same 1, 5, 6 proteins coming out every single batch, and supported with all our characterization studies, all our comparability studies, all our process development studies performed with LC-MS. We can show that our process is consistent, we have the same proteins, and maybe we even can show that these proteins are not either immunogenic or not affecting the product quality. So in principle, who cares about the number, the quantity number? If they are not immunogenic and they don't pose any risk, why do we need to quantify? As long as I can show that they are the same from time to time, and if there are any process changes, I will always use the LC-MS to characterize the process changes. So in the future, we shouldn't. It's not necessary to present a quantitative number.
In a way, it's an arbitrary number.
Yeah, it is.
On the analytical method.
Very much arbitrary number.
Can you make a guess? I mean, how far away is this future? Is this 10 years from now, or do you already have to think about it now, because the project you're working on, if they have a 5-10 year development time, then you need to think about it already now?
I hope that can inspire other companies to do the same. So if there are more companies having the same thinking, maybe the authorities will also accept. And the authorities, I mean, they are also scientifically based, and they are also risk assessment based. So they will always look, is there any problematic HCP? Have we shown during the clinical studies that the same proteins do not have any immunogenicity issues? So they will probably also accept our approach. I cannot say if it's 5-10 years, but I can envision a stepwise approach where at the end many more people also accept the LC-MS one single approach.
Yeah. So with that, thank you very much, Margarita, again, for excellent presentation and very good discussion. I think you have an insight that many people can learn from. So we don't have time for more questions now, but if you have questions, then please type them in the chat, and then we will get back to you by email. So with that, thank you again. Thank you for your attention. So bye-bye.